Dr. Larry Goldstein is wonderfully generous, and he is also going to be providing now a scientific keynote address. He's already been introduced, but I have to share a couple small anecdotes about Larry. He was one of the founding members of the Genetics Policy Institute to alleviation of human suffering and bringing stem cells into the forefront and translating this research into eventual treatments and cures. He was joined by Alan Trounson, who you heard from today, and Camillo Ricordi, who will be the director of the Diabetes Research Institute, Rudolf Janisch, who is our science keynote speaker tomorrow, as founding members of the Science Advisory Board. The fact that these scientists generously lent their names in support to a fledgling advocacy organization has made all the difference and is one of the reasons that we're all here at the World Stem Cell Summit today. Larry is also one of those gifted scientists that knows how to speak to the public. He is the author of a very famous tome, Stem Cells for Dummies. And for all of us who are sometimes forget what we learned in 10th grade biology, such as myself, that is an invaluable reference. So I'm very delighted again to bring to the podium Larry Goldstein.
All right, so just when you thought it was safe to return, I'm back. So I think, again, my job is to try to finish by about 11:30 so we can hear Andrew Grove talk about high stakes in the biotechnology and IT industries and how they relate. I thought, though, I do want to thank Bernie Siegel for inviting me to this, along with the organizers who invited me. Being involved and knowing Bernie for a great many years as I have, I've been continually amazed by his just relentless energy and enthusiasm for this area and the contributions he's made. I have to say, though, the thing that impressed me the most about Bernie is he's the only lawyer I know who's ever sued an alien cult. Some of you may or may not remember this, but there was a period about 10 years ago where the Raelians were marching around in Star Trek suits saying that they had come from some other planet or star and they were going to reproduce themselves by cloning and it was going to be free love for everybody. Those were quite the claims, but Bernie, in just a massive stroke of legal brilliance, sued them for custody of the child, recognizing that there was a real question of paternity under Florida law that really had to be resolved for the welfare of this supposed clone child. And I think Bernie's efforts helped to scuttle the ship of these folks who were clearly just total fruitcakes. So thank you for that, Bernie. It's a rare distinction. So I want to continue talking about how stem cell science and research is going to, we hope, lead us to improved therapies for terrible disorders. In the previous session, I talked briefly about our work on Lou Gehrig's disease and our determination to get this to clinical trials in the next several years. In this session, what I want to talk about primarily is the problem of Alzheimer's disease, and then as a parallel to that, how stem cell technology is letting us begin to understand how each individual's unique genetic variation may lead to disease risk, which I think is not only a very interesting scientific problem, it's a medical problem of enormous impact, as you'll see. So, reorganizing again. Here we go. So I'll return again to this problem of neurologic and neurodegenerative diseases, which include, of course, Alzheimer's disease, Parkinson's disease, Huntington's disease, and ALS, and a number of others. And as I'll illustrate for you momentarily, the frequency of these diseases in the population is simply enormous. And as a result, there is tremendous medical need. And the problem that we face as scientists trying to develop better therapies is there's a real shortage of what I would call rigorous therapeutic hypotheses. And what I mean by that is to again return to the notion of a broken engine, where in order to fix the engine, you have to have some guess, some idea, some logical idea about what's wrong with the engine so you can go in and effectively fix it. And that's true in therapy development as well. The more you understand about how a disease works, that is, how a disease results in a change in normal biology, the more likely you are to be able to develop an effective therapeutic intervention. Now, the problem in the neurologic and neurodegenerative disease area is we really have a very small level of that kind of understanding, partly because we don't fully understand how the brain works, and partly because we don't understand how that normal brain function fails in diseases like Alzheimer's. And the result at the moment is we have few, if any, effective treatments for these very common disorders. We have some symptomatic relief in the forms of drugs, but by and large, we have no therapeutic approaches that change the course of disease, as you'll see momentarily. Now, the problem that I'll focus on in particular is this problem of Alzheimer's disease, which, like Lou Gehrig's disease, can be described relatively simply. So let's look at what this disease is. So first of all, it's really common. So it turns out that 10% of people over the age of 65 have Alzheimer's disease. 50% of people over the age of 85 have Alzheimer's disease. And I suspect that most people in this room share my own desire, which is to productively live to the age of 85, still working and still being a productive member of society, and not a burden on my family and friends. The disease is progressive. Once it's diagnosed, that's it. It just proceeds inexorably, and of course that means that it's incurable. And there really are only a few drug therapies that have any effect on symptoms. The standard one are so-called cholinesterase inhibitors, which slightly improve memory and understanding, but they only work for a few months, and they have side effects that make it hard for some patients to tolerate them. There's some evidence that a drug called memantine may also have some slight effect, but none of these drugs alters the basic rate of progression of the disease, and no treatment yet changes the course of the progression of disease. Now, I said that the disease is simple to describe in some ways, and that is summarized here. Primarily, Alzheimer's disease, like many other neurodegenerative diseases, is a disease of what we call synapse loss. What I mean by that is the way brain cells, neurons, send signals to each other throughout the brain is at so-called synapses, which are areas of close contact between brain cells, and that's how signals are transmitted from one brain cell to another to form the circuitry that allows us to move and think. If those connections are disrupted, brain circuitry no longer works properly. And that's what happens in the early stages of Alzheimer's disease. You lose the connectivity between brain cells, and then after those synapses are lost, it eventually progresses to massive death of brain cells and the atrophy that you find in people who have been afflicted with this disorder. Now, the cause for almost all Alzheimer's disease is a complete mystery. In 99% of cases, we don't know why people developed the disease. But we do know that there are characteristic pathologies that you can find in the brains of people who've perished from this disorder, and they are the so-called amyloid plaques, which are found in large numbers in the brains of people who've died of Alzheimer's disease, although as you'll see, you often find large numbers of them in the healthy elderly. And then there are also the so-called neurofibrillary tangles, which is another kind of aggregate found in the brains of these people. The current dogma in the field is that something about these plaques perhaps their constituents known as A beta, cause all of the downstream biochemical abnormalities in brain cells that ultimately lead to tangles, synapse loss, and neuronal death. And this dogma is referred to as the amyloid cascade hypothesis, and there are biochemical features of this hypothesis shown here, namely, jargon alert coming here for those of you who are not hardcore neurologists. A beta is predicted to lead to activation of this thing called GSK3, which leads to phosphorylation of the proteins in these aggregates. So that's the prevailing wisdom or dogma, as you want to look at it. Now, I'll just point out that to some small minority in the field, of which I am a member, there appear to be problems with this accepted hypothesis. First, and most important perhaps, is that this view, although billions have been invested in drug development based on this hypothesis, no effective drug has yet come out of the drug development based on this idea for what's going wrong. And that's a real problem. And partly because, I'll just use a little more jargon here, the dogma for what's going on lacks what folks like me think is a credible molecular mechanism for what's going wrong. It doesn't have the level of detail and evidence that's needed to persuade hardcore skeptics such as myself that this is what's really happening. It's also been a bit of a moving target. That is, as it has failed various types of experimental tests, rather than disqualifying the hypothesis, it has often been adjusted slightly to fit the new data, which is something that I have a certain amount of qualms about. And perhaps the biggest problem is, with all of the concerns and reservations about mice, nonetheless, when you make mouse models of Alzheimer's disease, that is, mice with large numbers of so-called amyloid plaques in their brains, they don't have massive neuronal death. They have a modest amount of synapse loss. Now, you could raise the argument, well, can we really test if the mice can find their car keys, or can they find their way home after going to the pharmacy, or things like that? And there is a legitimate concern about studying memory and behavior in mice. And of course, mice that live in the lab are, how shall we say it, dumb as posts. And so it could be that when you give them, quote, Alzheimer's disease, they have nowhere to go. They're about as dumb as they're about as demented as they can already, I'm serious here, that you have no way to measure loss. But given that, the problem is we really haven't been able to find a therapy based on this approach. And it turns out, as Arnold so beautifully described, there are some real differences in the basic biochemistry of human brain cells and mouse brain cells. And in disease, that may really be important. The details matter. Okay, so what are we trying to do? Well, it turns out that although there's a primary hypothesis that is favored by most workers in the field, there are actually a number of other, in my view, credible ideas about what's going wrong in this disorder. The various ideas are based partly on experiments in mice and partly on observational studies in humans. And that ultimately illustrates one of the problems with making progress on a human brain disease. So if you think about it, if you're a hardcore molecular biologist and cell biologist such as myself, where I like to take mouse brains and grind them up and study all the molecules in there and how they interact and the like, how am I going to do that with people? So my friend Evan Snyder just walked in down here, I see. So I'm going to do an experiment on Evan. I want to study human brains. So, Evan, I have a drill in my lab with your name on it. Are you willing to come on down tomorrow afternoon and let me drill a small hole and just take a few grams out for some experiments? Absolutely. The last person to take me up on that was a community college student. Now, I'm not sure what that says about Evan. All right, all kidding aside, you see my problem. Not only that, I don't know whether Evan is going to develop Alzheimer's disease or not. So I don't know whether what I see when I grind up chunks of his brain, if I could even get them, is going to tell me anything that I can relate to disease biology or not. It's a complicated problem. Maybe he'll develop it in 20 years. Maybe he's in the early stages. Maybe we didn't notice and he's actually got it already. I don't know. So the question is, how can we make progress on this disease, which in many ways has uniquely human features, using stem cell technology? Okay, so, and of course, how can you test ideas, and then how can you develop drugs? Because that's the name of the game here. Okay, so here's what we've been trying to do, which is we've been using human embryonic stem cells and so-called human reprogrammed stem cells, which were described in the previous talks, to try to make human neurons, human brain cells in a dish that will develop the biochemical features of Alzheimer's disease, and then use that to test ideas and look for drugs. And so let me tell you how that's going. Now, one of the first problems, and this has been alluded to, is when you make different types of cells, starting with stem cells, you need to have some way to know what those cells are once you've generated them in a dish, because looking through a microscope at a cell doesn't always tell you everything you need to know about that cell's function. And also, if you want to do what I would call rigorous, careful biochemistry, you sometimes need to be able to purify the cells so that you're not trying to study a mixture. Imagine if you were trying to study the properties of red M&Ms blindfolded in a mixture of M&Ms of all colors. This is something of the problem we face. Okay, so it turns out that it is relatively straightforward, and has been for some time, to make these types of cells, shown here with these long fibers, which are actually human neurons, human brain cells, starting with embryonic cells or reprogrammed cells. But the problem is making them consistently and in purified condition. And so we've collaborated with a private sector company, Becton Dickinson, to develop so-called surface markers, that is, molecules that are on the surface of different types of cells that we can then attach other molecules that have colors, red or green or blue, that only attach to some cells of one type and not others. And ultimately, what that lets us do is to develop a so-called signature of surface molecules that we can then purify using fluorescence-activated cell sorting, so that all of the cells that have green molecules on their surface end up in one tube, and all the cells with red molecules end up in another tube. And if you do that properly, you can get what's shown here, which are populations of cells at reasonable quantities, not yet at full scale, but at quantities suitable for some kinds of biochemistry, that are 90 or 100% pure neurons. They have the electrical properties of functional, well-behaved neurons. That is, they have normal capacitance, they have normal action potential generation, a number of other electrical properties that tell us that they're well-behaved neuronal cells, and then we can go ahead and study their properties. Okay. So, here's an experiment that one of my graduate students did in the past couple of years. So in this case, what we're going to do is we're going to start with so-called skin cells, which are really a special cell called a fibroblast taken from a skin biopsy. And the work of Shin Yamanaka some years ago and a number of other labs since then has made it relatively routine to treat those cells with genetic factors carried by retroviruses. And those genetic factors allow us to generate so-called cultured pluripotent stem cells. What I mean by that is cells that can make every cell type in the human body, but because we've used this so-called reprogramming mechanism, these cultured cells have the same DNA, the same genome, as the person whose skin we biopsied, so that these things are all more or less genetically identical, the person, the skin cells, and the cultured pluripotent stem cells, although if there's time, I'll come back to a caveat about that. And so you can see the idea. What I said at the beginning is that in 99% of the cases, we don't know why people who develop Alzheimer's disease developed it. But in 1% of the cases, or maybe a little bit less, we do know why they developed it. These are so-called familial or hereditary forms of Alzheimer's disease where well-understood, well-characterized genetic changes lead to so-called early onset Alzheimer's disease. That is, these people develop Alzheimer's disease in their 30s and 40s. Really nasty. And so the notion is, if you could make these so-called reprogrammed or iPS cells from individuals who carry these genetic abnormalities, you could ask the question, when you make neurons that carry those genetic abnormalities, do they exhibit the biochemical behavior expected of Alzheimer's disease neurons in the lab in a dish? And then, can you use them to test ideas and screen for drugs? So that's one path forward. There's a second path forward, which is much more speculative. And that comes from thinking about the 99% of Alzheimer's cases where we don't know what caused the disease. Now, a couple comments about that. First of all, even though most so-called sporadic Alzheimer's disease is not hereditary in a simple fashion, it is more common in some families than in others. And when you look at identical twins, the chances that they're concordant, that is the same for developing disease, is pretty high. And the estimates are that about 60 to 70% of the risk for developing Alzheimer's disease is actually caused by genetic factors, the individual patterns of variation that distinguish every one of us in this room from the other. We all have different susceptibility to different sorts of disease, and a lot of that is encoded in the variation in our DNA, in our genomes. And that's true for Alzheimer's disease as well. It is also clear that there are environmental influences on the development of Alzheimer's disease. And interestingly, the best evidence for an environmental cause comes from the study of World War II war veterans, where it's become apparent that so-called traumatic brain injury, head injury, is one of the major predisposing environmental factors for the development of this disease. And I'm sure many of you have followed the developments in the NFL over recent years, where concussive injuries are leading in some of the ball players to pathologies in their brains post-mortem that are reminiscent of some of the changes seen in Alzheimer's disease. And this is a very important and developing story. But I want to just focus now on what we can learn about the genetics, the variation patterns. And with sporadic disease, the way you could imagine using this reprogramming technology is summarized here. So imagine that we had a bunch of patients who developed sporadic disease, and we biopsied them to get their skin cells, and then reprogrammed them to make stem cells. And what we've now done is to capture the genome of each one of those people in a stem cell line that we can then use to make brain cells in a dish. So if I did that to Evan, instead of making a hole in his head, which he might let me do, I'm surprised to hear, I could biopsy his skin cells in his arm. Much more likely I'm going to get to do that. I can make then stem cell lines that have Evan's unique genetic variation, and I can study them. Now, obviously I still don't know what he's going to develop, which is why I'd prefer to start with patients who have already developed Alzheimer's disease rather than waiting around to see what happens with Evan. And so that's what we've been doing. And again, the question is, do any of those brain cells develop biochemical patterns of behavior that look like Alzheimer's disease or what we see with hereditary mutations? So here's the experiment that my graduate student, Mason Israel, did in the past few years. He started with a very small sample because the technology is incredibly labor-intensive at the moment. Half a dozen patients, 2 that were controls with no sign of dementia in their 70s or 80s, 2 patients with sporadic disease, and then 2 patients from a family in Finland who had a genetic mutation that caused Alzheimer's disease in their 40s or 50s. Very virulent form of disease. He made skin cultures from all of these patients. He made multiple independent reprogrammed lines from each patient. That way, if there's any experimental artifact in one of those lines, we get around it by having multiple different representatives. He then made neurons from all of these different stem cell lines, and then he studied their biochemistry. Okay. One of the great things about being a so-called faculty member or principal investigator is I get to say the royal we a lot when we did these experiments. In fact, this student did an enormous amount of work on the characterization of these cell lines and their behavior. Months and months of work become these little tables that go in theses and publications, and so all of these lines were tested for genetic normality, pluripotency, and a number of other properties that were important, and all the neurons that were generated were tested for a number of properties that rigorously established that these are well-behaved, functional neurons that we've generated in our dishes in the lab, and then you can begin to develop methods to measure their biochemical properties. And so I'll just give you a few examples of this. I mentioned at the beginning that there is this so-called A-beta peptide, which is a major constituent of these amyloid plaques that you find in the brains of people who develop disease. And so if you look at what these neurons in a dish are generating using some very sensitive methodology, what we see is that the control neurons make very little. The neurons from the hereditary forms, which we expected should make large quantities of these peptides, in fact do. And interestingly, one sporadic patient's genome does not generate neuronal misbehavior, but one sporadic patient's genome does, interestingly. Very similar to what we see with this known genetic mutation, even though this patient doesn't appear to carry that genetic lesion. You can then look at so-called activation or aberrant activation of this kinase, and again, the 2 hereditary patient neurons have this abnormal biochemistry, as does one of the sporadic patients, none of the controls nor the other sporadic patient. And then when we look for aberrant phosphorylation of this protein found in neurofibrillary tangles, the same pattern. The 2 hereditary patients and the sporadic exhibit this behavior, the 2 controls and the other sporadic doesn't. Now, that's a very small sample. It is really early in the game. But these findings tell us things that I think are very important. So first, it tells us that even though these neurons in the dish have not been aged for 30 years, you'll see that a 30-year experiment is a little bit incompatible with graduate school as we know it. So, nonetheless, they recapitulate early features of the abnormal biochemistry of Alzheimer's disease. And that's really important because it means we may be able to use them to make progress with a truly human version of disease. Second, there is a prediction of the dominant hypothesis, which has to do with the so-called proteolytic processing, otherwise known as cleavage of this protein into pieces, and how that relates to the downstream biochemical changes. And the interesting thing is that one of the most important predictions of that dominant hypothesis is not upheld in these neurons. In fact, it looks as though a slight variant on what people think is going on may be more important, and that may give us a path forward that we have not been able to achieve and develop based on studies in animals. Third, a real surprise is that one of these sporadic patients has a genome that generates neurons with unusual behavior. That potentially gives us, for the first time, a way of studying sporadic disease using this sort of genetic trickery, which is something we've never been able to do in animals. Now, before we celebrate, remember, this is a very small study. We need to do literally dozens more patients before we can be sure that this is meaningful for the large number of sporadic patients that we encounter. But at the very least, it tells us that it's possible that the pattern of variation can generate Alzheimer's type behavior in purified human brain cells. And if this idea is correct, it may give us a way of doing early genomic-based diagnosis in the coming years. But that's something that we have to test very carefully before we know it's true. And then finally, this, for the first time, gives us a way of using true human neurons with aberrant Alzheimer's type biochemistry for screening for drugs, which is something we hope to initiate within the next year. So stay tuned. So let's skip this particular issue, and I want to close on this final idea. In talking about sporadic Alzheimer's disease, I described for you what in some ways is the biggest problem in human disease biology, which is trying to learn how each of our individual genetic variation leads to a number of behaviors relevant to disease. I mentioned that we each have different disease susceptibility because of our unique pattern of variation. When you go to a physician who asks about your family history of disease, that's exactly what you're being asked about, is what kinds of diseases run in your family because of your patterns of genomic variation. You also probably know that every one of us has individual, actually somewhat idiosyncratic responses to different sorts of drugs that are used to treat diseases. A drug that I tolerate well might not be tolerated well by Bernie or Evan or somebody else because of the genome and because of the pattern of response encoded in that genome. And so we've been trying to move now beyond the sequencing of the human genome to try to harness stem cell technology to help us understand in an experimental way how genetic variation leads to these different sorts of cellular and biochemical behaviors in drug response, in disease, in response to injury, what have you. And that's led to the initiation of what I sometimes call the Craig Venter Project. So Craig is my friend and neighbor in La Jolla. He's shown here. Actually, he grew a beard recently. Looks a lot better, I think. And so here was the idea. Craig was one of the earliest human genomes that was completely sequenced with every known genetic variant that might increase his risk to different sorts of disease identified. And from my personal standpoint, Craig has a bunch of really nifty risk variants that predispose him to Alzheimer's disease. And so, now, by the way, Craig is not so thrilled about that, but he has been very open about this and, in fact, published his genome sequence in the scientific literature with the analysis of his risk variants. So what we did was we tracked Craig down one day and biopsied him to generate skin cells, and then we reprogrammed his skin cells to this cultured pluripotent state. And we've been making, in a dish for a number of months now, Craig's neurons. Now, Craig would really prefer it if we would take those neurons and transplant them into his brain. We're not going to do that. We don't know how to do that safely, and it might make him even more difficult to deal with. So it's not necessarily what we don't want to do any harm here. But what this lets us do now is study Craig's neurons in a dish and ask the question, using other advanced genetic technology, how do these risk variants in his genome, how do those affect the behavior of his nerve cells? Because we can manipulate those. We can make more of them, we can make less of them, we can change their character, and study in his unique genetic background what's going on. Now, one other thing sort of fell out of the analysis of Craig, and this is a good news, bad news story. With my collaborator at the UCSD Bioengineering Department, Kun Zhang, and his graduate student, Atharva Gore, because Craig's genome had been sequenced, we could compare the genetic integrity of the genome of his reprogrammed stem cells. And what we discovered is that somewhere during the reprogramming process, or pre-existing in his skin cells before we biopsied him, there were a number of genetic changes that were not present in his so-called germline genome. That is, something about the methodology had induced genetic misbehavior, which is a problem. The frequency is about 10 times what we would like it to be or expect it to be, and we've been able to pin down that about half of these variants, that is, these genetic changes, occurred in his skin cells, and about half occur sometime afterwards during the reprogramming process, we think. Now, this is not just unique to Craig. It turns out that we've looked at reprogrammed lines made from multiple donors in multiple labs, both by viral-based methods and by so-called safe methods, and they all have this feature. And we think, but we don't know yet, that many stem cell lines, including mesenchymal and others, may have these sorts of genetic lesions, and I think Nissim Benvenisti, who's here, may tell you about his work on some of these issues. But the point is, when we published this, we got a lot of concern. Does this mean that reprogrammed stem cells are too dangerous to use for therapy? The answer is no, that's not what that means. It means that we've discovered a feature of these cells that may have to be monitored during the development of therapeutic approaches. It's not bad news because, in my view, every problem you identify before you go into clinical trials in humans, that's a step forward because it saves you from a potentially catastrophic outcome. Things that you know about are things that you can control or at least potentially manage. And so that's why this is ultimately important. And genetic integrity may become an important part of how we QC stem cell lines moving into the future as we think about using them more and more for therapies in humans. So at the beginning, when the organizers were welcoming you all to this meeting, they all gave plugs for their local institutions, and I am certainly not above that. One of the reasons I moved to San Diego 15 years ago was because of the problem that I've summarized here. When I was a faculty member at Harvard and when I spent my training years at a variety of other institutions like MIT and the University of Washington, I discovered that most departments were like silos, where the analogy is, I was a grain of corn in one of these silos, and I never got to talk to any of the grains of wheat or grains of, what else would there be? Rice, I suppose, soybeans, in the other silos. Whereas San Diego turns out to be a place where there's an enormous amount of communication between scientists, not just within a discipline, but across disciplines. And that's really important for a number of reasons. I hope that you've gotten the sense from what you've heard this morning that the problems in human disease, while they are ultimately solvable, are really tough. And they require teams of scientists and physicians with different sorts of backgrounds and training to work together to solve these problems and make progress as rapidly as possible. And this is what we're trying to do in San Diego at the Sanford Consortium for Regenerative Medicine, which, as you've heard, we're going to occupy very shortly. So let me close by thanking a lot of folks who helped make this work possible. In particular, this student of mine, Mason Israel, a number of collaborators on the generation of human stem cells, human neurons, and their characterization. And of course, Craig Venter, who let us biopsy him, as well as the many other patients with Alzheimer's disease that we've been studying. Thank you.